So in this part, I'll mostly just focus on the structure part, and in the next lecture, I'll try to talk a bit about the kind of work people have done to understand function in development and how it relates to structural analysis. And for most of the examples, I'll stick to C. elegans, which is a nematode. It's a famous model organism. I'm sure you must have heard about it in the previous lectures. It's a small system, so it's very convenient to work with. So far, most of the workshop we have been focusing on the structural information given the connectome, right? So you all must be familiar with the adjacency matrix by now. So if you have a connection, a network like this, if two nodes are connected, the corresponding row and column entry should be 1, otherwise it will be 0, right? Now adjacency matrix generally has, it's enough, it explains the connectome completely. You have all the structural information in that matrix. Now the first natural question to ask is, given this connectivity, what can you say about function? And usually it's not very trivial because you need some additional context to make sense of function. But a good starting point is to look at some relevant dynamical models and see how the dynamics plays out on the network, right? So these are some examples of dynamics that you can think of on a network. So you can have interacting spins in a network. So every spin can have 2 states. You can think of them as a magnet which can point up or down. And depending on the interaction with the neighbor, they'll try to align if they have ferromagnetic interaction. So you can study properties like what is the magnetization of the system. Given that they're interacting, their interaction is constrained by this network. Magnetization just means the overall orientation. This is also, a very good toy model for opinion dynamics. So if you're given some opinion, if you ask about what opinions people have about a particular topic, it can be represented as a 2-state, system. Like maybe they like something or dislike something. And if they're friends with someone, they'll try to align. If they have enmity, they might tend to anti-align their opinions. You can also think of oscillator models. So individually, you have these oscillators which have their own intrinsic frequency. But once they're coupled to other oscillators in the system, they might end up synchronizing. And I think one good familiar example would be when you applaud after a performance. Initially, we all tend to clap with our own natural frequency, but over time we tend to sort of synchronize, right? But even though these dynamics are very different, and the tools that people generally use to study these systems are very different, in some limit you can reduce it down to a diffusion-like process. Now, if you're not familiar with diffusion or if you don't have intuition for it, just think about random walkers on a network. So the dynamics of random walkers on a network or diffusively coupled systems can capture the behavior of these two systems up to some limit. That's the key part. It captures it only up to some limit. And but before we go to networks, we can just talk about diffusion in physical space. So if you think about a one-dimensional lattice, and let's say there is a particle or a person which can randomly hop left or right with equal probability, right? So you can write down a probability equation for this system. You can ask, what is the probability that this particle is at a particular position x at time t plus delta t? And this depends on two. So the way you can reach here, there are 2 ways basically through which you can reach here. Either you were at x minus delta x in the previous time step and you took one step forward with half probability, or you were at x plus delta x and you took 1 step back with half probability. So you can write down this evolution equation. If you rearrange the terms, you can show that the change in the probability density is given by this expression. Now this particular term, minus 2 px, sort of reflects the fact that you have 2 neighbors in the lattice, right? This is where the information of neighborhood is coming in. And you can simplify this equation and show that it reduces to this particular expression. So this constant, diffusion constant, is basically dependent on the lattice spacing. So it goes as del x square, and it also depends on the time increment. So it goes as 1 by del t, right? And the double derivative, we can also represent it using this Laplacian operator, right? This is just a notation. So the one way to think about it is to think that this operator is acting on this density, the vector of density. So, and intuitively that equation, is there, I mean, most of you are familiar with this equation, right? Is there anyone? Okay. I mean, intuitively you can think of it like this. If this is the physical space and this is the density, let's say at some arbitrary time, these are the points where you have huge curvature, right? And this is where the double derivative d square p by dx square will be negative or positive. So if it's a peak, it will be negative. If it's a valley, it will be positive. So dp by dt just tells you that this equation will tend to flatten out the curve, right? So if you're at a peak, you'll go down, and the other way around. So diffusion, intuitively, you can think of it as a homogenizing force, at least for this talk. Now, if I ask you that, let's say there is a random walker who starts at the origin, and he's just randomly hopping with equal probability. After time t, what is the expected displacement from the origin? What do you think that, how would it depend on time? I think some of you may know the answer. If I look at the mean square displacement, right? So if there is a random walker who starts at the origin, and at every time step, he is making a hop with equal probability. After some time t, you would expect the random walker to be somewhere, right? It's very unlikely that, I mean, it's not guaranteed that he'll be at the origin. So what is this expected displacement? How does it depend on time? I mean, you don't have to solve it really, you can. Okay. Does anyone know the answer? Okay, at least tell me this. If this is time, would it vary linearly? Would it grow faster than linearly or slower than linear? Linear. Who thinks it's linear? You have to make a choice. You can't be neutral. Pick a side. Is the question clear? Okay, let's take another scenario. Let's say there is a particle which starts at the origin and it's moving in the same direction with constant velocity. Now, what will be the expected deviation? How would it vary with time? Linearly, right? But now this random walker is trying to move back and forth probabilistically. So would it be greater than, less than linear, right? And you can sort of figure it out just by looking at the expression for diffusion constant. So if you just rearrange those terms, you can show that del x goes as d into del t to the power half. So it grows slower than a linear dependency. And that's important because in biological systems, often when things rely on diffusion, it can't operate on a long scale, right? It's not feasible. So you can do a very similar thing for the, okay, you should ask questions. I know it's obvious, but I can't tell if you are following or not. So if there is any doubt, interrupt. Just like we wrote the evolution equation for the one-dimensional lattice, we can do it for any arbitrary network. So if you have an adjacency matrix A, we can write down the probability change at a particular point as the sum of influx. So basically, what is the density of random walkers in my neighboring node, and with what rate are they flowing in, minus the rate at which you flow out. So the density of random walkers at my node, sum over all my neighborhood. So if I have more neighbors, I'll leak out faster. Now, the sum of these neighbors is nothing but the degree of your node, right? Now, what you can do is you can define a new matrix called the Laplacian, where Lij is nothing but minus of Aij and Lii. So what is Aii? So if I take the first node, what would be A11 for adjacency matrix? 0, right? There is no self loop. But for a Laplacian, I can define it as minus Ki, the degree of the node itself, right? So this equation can be compactly represented in this form, right? I've somehow incorporated these two terms in the same matrix, and this matrix is what I'm calling the Laplacian. So if you look at the adjacency matrix, all these diagonal terms are 0, and you have some terms here, right? Now you can replace the diagonal terms with the negative of the degree, minus k2 and so on. And the negative of this is Laplacian, right? And even here you can see that just like we had a term, this factor which captures the neighborhood of both these points, the diagonal entries of the Laplacian captures this sense of neighborhood. So a lot of information about the structure and dynamics can be obtained from the Laplacian. Now, if your linear algebra is rusty, this is all we are going to use for now. If you have a matrix of size, let's say n cross n, and it acts on a particular vector of size n cross 1, right? Now, you'll get some new vector after this matrix acts on the vector, right? And you can imagine this operation where A multiplies with V to give you a new vector as just scaling, rotation and scaling in some space. So simple example is if you take some matrix x1, y1, you multiply it with, let's say, some matrix 5, you'll get a new vector x2, y2, right? So in xy coordinates, you can represent this as a vector x1 comma y1 and this as some new vector x2 comma y2, right? And this matrix will tell you how they are rotating and scaling. Now the claim is that for most matrices you can find some natural directions along which there is no rotation, there is only scaling. So by some factor, let's call it lambda. Now these particular vectors are called eigenvectors, and the scaling factor is what we refer to as eigenvalues. Now this is a very convenient space to work in. So if you want to think about the same operation using eigenvectors, what you can do is you can take this matrix x1, y1, figure out the eigenvectors of this matrix. Let's say the eigenvectors is along these two directions. You can write down this vector as the sum of two components along the eigenvectors, right? And then you can act the matrix on this sum instead of acting it on the final vector. And this is convenient because along the eigenvectors you don't have to do any rotation. You can simply scale it by the first eigenvalue and the second eigenvalue. And then you can add them up again to get the new vector. So if you want to solve an equation like this, what you can do is you can write down the initial vector p. So p tells you the density at every node. It's a vector, right? You can take the overlap of those vectors with all the eigenvectors, which is this term. You can rescale it by some factor, this exponential factor, I'll come to that, and it's along some particular eigenvector. So what you're doing is you're taking the initial density, projecting the components along the eigenvectors, evolving them independently by some scaling factor, and you're adding them up. And when you add them up, you get the density at some particular time t. And the time comes in through the scaling factor. Now, can you see why it would be an exponential? Why is the scaling factor this? So this is nothing but the solution to that equation, right? Right. No, come again. Right. No, you're right. So you're saying that if this was a scalar, you would just get p going as e to the power minus lambda. But how do you get the eigenvalues? It's the same thing. I'm just being nitpicky. So look at this equation, right? This is the definition of eigenvector. When the matrix acts on it, you just rescale the vector, you don't rotate. So you can replace this L acting on P as the sum of the rescaled eigenvectors, right? So if P is an eigenvector, L, P is some eigenvector, this will just give you PE divided by lambda, right? This is the definition of eigenvector. And then it becomes a scalar, right? Then what the argument you give will hold. And you can see that the lambda, the eigenvalue, is actually a time scale. It tells you something about the time scale. If lambda is very large, the eigenmode will decay pretty fast. If it's very small, it will persist for a long time. So the time scale of that particular eigenmode is given by the inverse of the eigenvalue, right? So what it's essentially saying is when a Laplacian acts on a vector, most of its eigenmodes are shrinking. They're going to 0. And you can go back to the system of interacting spins and a network of oscillators. And okay, let's take the spin case for now. So this was a model described by Anuran. This is a continuous Hopfield model. It's not exactly the continuous Hopfield because you have a more complicated functions, but this is like a simplified version. You have a node whose activity is decaying, but your neighbors will try to push you to +1 or -1. And if you just rearrange these terms, you'll get an expression where you can show that the evolution of these spin states depends on some function of this spin and a Laplacian-like operator acting on the matrix. So in some limit, this terms become dominant, and you can show that this essentially tells you all you need to know about the dynamics. Similarly, a very different system, like a network of oscillators, without any interaction between the neighbors, each oscillator is just sort of oscillating in its natural frequency. But you can introduce a coupling. So this is a famous Kuramoto model. And this coupling is, if K is strong enough, the coupling constant, you can get synchronization. But if you assume that the system is close to synchronization, you can do a tailored series and show that this interaction effectively boils down to a diffusion-like interaction. Now these are only true in certain limits. So if you actually look at these systems on a network, you get very rich behavior, and diffusion won't capture all of it, but it's like a first, a 0th order model, a good null model. So anything apart that you don't, any deviation from a simple diffusively coupled system will tell you something about the peculiar property of the node dynamics itself. Yeah. Right, so here I'm subtracting, okay, so I'm basically just doing this. Take the first equation and just subtract this and add this, right? Nothing changes, right? I'm subtracting and adding the same term. Is it making sense? Huh? Oh, okay, wait. You take the first equation and you add this term, Jij Si. It's visible, right? Then you subtract the same term, right? Nothing changes. And this sum is over J. So you can pull out Si. And what is this term? Sum over Jij? Ki. So you can absorb it into the first term. Right? I mean, obviously this will contribute to the dynamics, but the Laplacian still could tell you something about the, and it makes sense if you remember some of the previous talks where the speakers tried to, sorry, it makes sense, right? Huh? Sorry? Oh, sorry, this is sum over J, your neighbors, just your neighbors, right? Yeah. Yeah. Yeah, so if you remember in the last few days, some of the speakers, they tried to look at the Laplacian, and they were able to show that it somewhat explains the functional connectivity, the functional network, but it doesn't do good, I mean, it doesn't explain it all, but it does way better than, simple, correlation between structure and function. So it's like a first step towards understanding dynamics. This is just one example of a Laplacian. So if you take this matrix, you get the same off-diagonal terms with a negative entry as the adjacency matrix, but the diagonal entries have the degree, information about the degree. So this system has 6 nodes, right? So it will have 6 eigenvectors and 6 eigenvalues. Can you point out some eigenvalues or eigenvectors? 0, right? This was probably discussed. What would the eigenvector be? Right. So this is one trivial eigenvector that you get, and it makes sense because the sum of each row or column is 0. So if you multiply it with a vector of all ones, you'll just get 0, a vector of all zeros. And if you interpret it using the time scale argument, the 0th eigenvalue tells you about the time scales which persists for infinite time, at infinite time, right? So if you remember, time scales went inversely as lambda, 1 by lambda, right? So how do you interpret this result? Is there a natural way to interpret what the eigenvector is telling you? Okay. So if you, again, what is the Laplacian telling us? It's telling us about the dynamics of random workers on a network, right? So as t goes to infinity, what would you expect the density profile to look like? Homogenized, right? Because diffusion is a homogenizing force. So this sort of tells you the steady state distribution, right? I mean, this is not related, but if you try to simulate a random walk problem on a network, so let's say you have some network, and let's say at every time step, this random walker is picking one of its neighbors and just randomly choosing one of them and hopping to that side, right? If you simulate this system, or if you solve it, what do you think the steady state distribution would look like?
For a longer period, it tends to become
T going to infinity. T going time, infinite time, yeah. Yeah.
Every node will have an equiprobable distribution, equal density at all.
You should try this because it won't give you equal density.
And the one with the higher degree will have a higher number of visits from
Higher number of?
visits from walkers.
Yeah. If you simulate the system, the density would be proportional to the degree.
Yeah.
So why do you think the eigenvectors are homogeneous? Because at the end we're simulating a diffusion, random walk problem. I mean, it's okay, it doesn't matter, just think of it. Yeah, so is it possible to have multiple 0 eigenvalues? Right? What does it imply? Right, I think this was also probably mentioned. Okay. Now, if you want to think, let's consider a very particular case of modular networks. So we have a network with 2 modules, and there is strictly one connection going from one module to the other, right? Now let's say there is a random walker who is just sort of exploring the network. Intuitively, we all agree that the random walker would be trapped in this module for a very long time, right? Because there's only one connection going to the other module. And we also know at t going to infinity, it would have explored the entire system. So there is a characteristic time scale where this particle makes the first jump to the other module. So sort of like the first passage time. Now this time scale depends on the inverse of the second eigenvalue. So one eigenvalue would be 0 and the next eigenvalue would be a positive constant. So the inverse would tell you what is the typical time scale required to cross over. So again, it intuitively makes sense because if you take a random network with no modularity and if you arbitrarily partition it, you would expect the time scales to be very low, right? It would quickly jump over to the next module. But as you increase the modularity, this time scale would increase, right? Which means the eigenvalue, the second eigenvalue would decrease, right? So we already have a 0 eigenvalue and we have another eigenvalue which is tending towards 0 when the network becomes modular, right? And the moment you fragment it, which is the extreme case of modularity, both the eigenvalues will be 0, which confirms to the idea that if you have 2 connected components, you get 2 0 eigenvalues, right? And you can show this in empirical networks. So if you look at brain regions of cat and macaque, they do show distinct timescale separations for some of the eigenvalues. So this is an indicator of modularity in the network. Why do you think modularity is important? Like, is there for these networks, for brain networks? Modularity or time scale separation, what could be the, how is it useful? Different? You mean like different parts of the network, different modules? So you're saying that because the time scales associated with certain regions of the networks are much longer compared to the rest, there might be some dynamics for some modules, some other dynamics. Oh. Yeah, I mean, I don't know if there's a clear answer, but yeah. It at least shows that there's a separation of time scales that operate at the modular level and at the network level. For directed networks, things become slightly complicated because you can have two definitions of degree for a node. The procedure is same. You define an in degree or an out degree Laplacian, and yeah. So the eigenvalue still informs us the same thing. You look at the eigenvectors. The eigenvalues will tell you how persistent are these eigenmodes, for how long will they last, the contribution of that eigenmode. But the eigenvectors have a slightly difficult interpretation. In some cases, it tells you what are the target nodes.
In the third of the in-degree Laplacian, is it 3 in the diagonal?
Third, this one, right? Yeah, so this is node C. It has 3 in degrees, right?
But this has like the row has -1 or -1.
A, D, F. Oh, sorry, this should be minus. This should be -1. That's a mistake. Yeah, the eigenvectors have a slightly more. It's not very straightforward how to interpret them. Sometimes they give you information about the source and target nodes, and sometimes they give you information about flux balance locally. So if you have, let's say, lots of node coming in, but only one node going out, so you're sort of integrating a lot of signal. You're like a bottleneck in the network, right? Or the other way around. Maybe you're only receiving one signal, but you're broadcasting to multiple nodes. So these are, I mean, there are mathematically rigorous way of showing in which case what sort of networks will give you what, but that's just too hard to, I mean, at least I can't follow through. So I just use these simple examples to sort of build intuition. So if you take two extreme cases like a chain or a cycle, right? If you take a simple linear chain where you have a clear source and a clear target, the first eigenvector, the corresponding to the 0 eigenvalue, will look like this red profile. So it's just selectively picking out what the target node is, right? And the second eigenvector corresponding to the smallest positive eigenvalue, it picks out the precursor to the target node. So in some sense, the eigenvectors have some information about the flow of signal in this network. If you take the other case, other extreme, which is a cycle, a directed cycle, you get one eigenvector which is the homogeneous state for 0 eigenvalue, which makes sense because if there are random workers, eventually they'll just sort of homogenize over this entire ring, right? But you also have different modes of different persistence, right? So as the eigenvalue increases, right, so as this is arranged according to the eigenvalue, so as the eigenvalue increases, you get these profiles which have finer and finer structures, and it makes sense. So if you have a very fine profile for the density of random workers, after some time diffusion will sort of smoothen it out, right, over small length scales. But if you wait long enough, it will also smoothen it out over large length scales, right? So these are the modes that will die out faster. These are the modes that will persist for a longer time. And it could be a good exercise to just sort of build networks which are composites of chains and cycles and see how the eigenvectors vary, right? So we come to C elegans, and by now most of you know why it's a good model organism and all that. One of the advantages being that its connectome has been completely mapped out. And broadly, there are like 3 classes of neurons. The sensory neurons, which receives information from the environment, and they pass it on to interneurons. The interneurons, they do a lot of integration processing, and they pass it on to the motor neurons. And it's the motor neurons that controls the final activation pattern of the muscles, which can modulate locomotion and so on. And synapses, they work, the signal transmission works by using neurotransmitters, these chemicals called neurotransmitters. So if there is a particular neuron which gets depolarized, it will release certain neurotransmitters on the end of the process. And depending on whether the postsynaptic neuron has a receptor to bind to it or not, it might successfully pass on the signal to the postsynaptic end. Now, if this receptor, so once the neurotransmitter binds to the receptor, it can open up ion channels that might end up polarizing or depolarizing the cell. So, strictly it's the receptor type that tells you whether the synapse would be excitatory or inhibitory. But for most purposes, you can also look at the neurotransmitters. So, there are certain neurotransmitters which tend to be excitatory, some which tend to be inhibitory. But there are some exceptions as well. So, if you look at the Laplacian of the connectome, and if you look at the first dominant eigenmode. So, what I mean is the eigenvector associated with the second smallest positive eigenvalue. Now, these neurons have been arranged according to the processing depth, which comes from anatomical information and experiments and so on. You can see that the sensory neurons have the lowest components. The components associated with the sensory nodes have the lowest values. Interneurons tend to have slightly higher values, and motor neurons are the ones that have the largest value. So, it sort of reflects this fact that the connectome is feed-forward driven. Signaling in this network is feed-forward driven. Now, this is mostly just sort of an aggregate. You're looking at some aggregate property of the dynamics, right? You're not really bothered about how each node locally is behaving. You're just looking at overall flow at a network scale. If you're interested in the other end, where you look at motifs and neural circuits, dynamics is obviously one way to model these systems, but even from structural information,
How do you want to interpret this?
So, strictly it depends on how many cycles does the network have. So, the eigenvectors of the Laplacian, it tells you 2 things. One is the source-like property and the target-like property of nodes. And the other thing it mentions is the amount of bottlenecks in a network. So, are there nodes which integrate a lot of signals but pass it along to only a few nodes? Or are there nodes which takes input from only 1 or 2 nodes, few nodes, but they broadcast it to a lot of nodes? So, that's why you see a lot of these fluctuations as well, because it's constantly getting integrated and broadcasted. Plus, there are lots of feedbacks in this network. So, obviously, you won't get a very clean, neat picture. But on an average, you can see that trend, right? The fact that it's feed-forward driven.
There are multiple colors. It's not just pink, green, and blue.
Right. So, C. elegans being such a small system, there are many neurons which are polymodal. So, these neurons, they act as interneurons, but they're also motor neurons.
So, when you're calculating the dominant eigenvector
Yeah.
of each of the nodes, you are considering those nodes in both the sensory, in both the motor and the
You mean this average?
Yes.
I don't remember, but it doesn't matter. Like, you'll get the same trend. Even if you exclude or include them, it's more or less same. And you can also, maybe I'm seeing too much, trying to read it too hard, but even the polymodal ones between inter and motor neurons, they're somewhere in between, right, in terms of values. These are sensory and motor, and there are also some sensory and interneurons, I think. But yeah. So, you look at the smallest positive eigenvalue, right, and there will be eigenvector associated with it, right. Now, C. elegans has like 300 neurons. We are looking at the somatic nervous system. We are ignoring the pharyngeal part. So, there are 280 nodes. So, the eigenvector will have 280 components, right, because your matrix is of size n cross n. 280 cross 280. These are the nodes associated with the sensory neuron. Right? These are the interneurons and motor neurons. They have been arranged, it's a convention which some people follow.
So the component means the 1280 eigenvector.
Yeah, we're looking at one eigenvector. So if this is my,
Oh, okay, okay, okay. Yes, sir. Yes.
right, there are n number of eigenvalues, we're looking at the smallest, the dominant one. Yeah, so if you're interested in dynamics at a circuit level or motives, then I mean, instead of solving dynamical systems, one crude approach could be to look at the overall sign balance in the network. So this concept of structural balance is essentially tells you how is the network designed, what sort of dynamics can you expect from this network given the nature of interactions. So let's say you go, so what you do is you go over all multiple loops in the networks, you take a product of all the signs, right? And if this product is positive, you say that the network is balanced in case of directed network, or if it's undirected, you say that there is coherent signaling. And you can sort of intuitively see why, because if you assume spin dynamics on this network, right? If there is a spin pointing up, this spin would also want to point up, right? Because there is a ferromagnetic interaction. And this spin would want to point down because of this anti-ferromagnetic interaction. And it's consistent, right? All three interactions have been consistently satisfied.
So can this be analogous to inhibited?
It's a bit more complicated, right? Because look at this triad, it has two inhibitory nodes, but it's structurally balanced. But this one has only one inhibitory node, but it's frustrated. Because if you assume that there is a spin up here, and there is a spin up here, because they're ferromagnetically interacting, this is an ambiguous state, because you're getting mixed signals, right? So these sort of triads will refuse to, I mean, the states would refuse to settle, right? And similarly for directed networks, what you can do is you can look at cycles, or you can look at semi-cycles, meaning these are loops, but they don't have the cyclic order. So here you have a source and a target, but the structure forms a loop, so you call it semi-cycle. But the principle is same. You take the product of all the signs. If it's positive, it leads to coherent signaling. So generally, if you want to fix attractors in your network, you would prefer balanced triads or coherent signaling. So if you remember the Hopfield formalism, so if you want to store a vector S1, S2, Sn, what you do is you take this vector and maybe I should. Yeah. So if you have a vector, let's say +1, -1, whatever, you can take its outer product and you can form a matrix, some Aij, and the Hopfield formalism tells you that this network will store this particular state, right? So Aij is nothing but sigma I, sigma j. Sigma is the state of these components. Now by construction, these networks are structurally balanced, right? Because if you take three nodes I, j, k, the fact that you have constructed it using this formalism ensures that the product over the triad would be positive, right? You can work it out. So balanced systems usually allow you to store patterns. It gives you stability. Frustrated systems can give you cycles, chaos, even just random, incoherent, persistent behavior. And usually you need some kind of sign balance to see persistent behavior in these networks. So if there is too much excitation, everything is just constantly firing or constantly on. If there is too much inhibition, the activity just dies down. But at a sweet spot, you'll see the activity somehow being able to persist itself. And when it comes to sensory and motor neurons, you need the context in which they're interpreting the inputs or what sort of activation patterns are being made. But a lot of the interneuron circuit, maybe these sort of features are more important. What is the persistent activity? Is it able to sustain activity even without external input? So in the case of C. elegans, there is a lot of adaptability in the sense that the cells can tune their neurotransmitter expression. So they can even switch between multiple neurotransmitters. And the postsynaptic neurons, they can modulate the receptor density. So you can modulate the sign and the strength of these interactions. There's a lot of adaptability in terms of how these networks are designed. In fact, to the point that it becomes very overwhelming because a lot of connections in C. elegans is very mixed. So it becomes a big question as to how is it able to modulate its interactions to optimize between this stability and plasticity, this adaptability in its behavior. Now, I've been avoiding dynamics for now, for the most part, and you can have many parameters which regulates dynamics. So, for example, the strength of signals, the input that you're getting. There could be thresholds of the neurons which can make a difference. But the one that I find the most interesting is time scales. And there is a huge variation in the time scales as to how this connectome processes information. So usually you have these chemical synapses which are fast, but there's also a lot of variation in its time scales. Then you have gap junctions, which Abhishek talked a lot about. So there could be two neurons where certain proteins called annexins, they come and form a pore-like structure. So when one neuron is depolarized, there could be a direct flow of ions to the other neuron, and these are usually very fast. But you could also have wireless connectomes. So maybe there are neurons which they just dump neurotransmitters in the extracellular matrix, or maybe there is some advective fluid and it reaches some neuron that is far away. So even without a hardwired synapse, there could be communication.
When does it take such a mechanism?
Huh?
Which kind of functions suit that?
So usually these are neuropeptides, and usually they couple to I forgot what it's called. It's called G protein coupled receptors, right? Yeah. So these usually play a role in modulation. So modulating the sensitivity of the neuron and among other things. Yeah. So for example, and this is also reflected in behavior in the sense that there are some scenarios where you have to respond really fast. So if you take a agar plate with the worms in it, and if you just tap the base on which it's kept, you'll quickly see that almost immediately all the worms will sort of retract their head as a reflex. Now this can't be a slow process. It has to be timed. It has to be quick. And there are many other processes where, so for example, if they sense some toxins, they'll quickly do a sharp omega turn, right? There are also many responses which operates in the timescales of minutes. So if you put them in nutrient-rich environment, they would sort of slow down and tend to stay in that niche because they don't have a bird's eye view of the niche. So if they encounter a nutrient-rich environment, they would preferably slow down just to stay there and maximize the likelihood of being around good, nutrient-rich environment. Yeah. There are also other behaviors, but it doesn't matter. The point is these behaviors are modulated across different time scales, and probably this reflects the time scale separation in the structure. Sitabra discussed this, so I won't go into a lot of detail. But before we talk about function or development, which often requires more context and more details, we can ask very broad questions like, is there something which the connectome is trying to optimize, or why is the connectome designed the way it is, right? So one simple assumption for nervous system could be that these connections are made to I'm getting conscious. The connections are made to sort of communicate efficiently, right? So it's obviously not true for sensory neurons and motor neurons because they need high specificity. Stop doing that. High specificity, but interneurons, you could sort of argue that their main task is to communicate signals across, right? So one easy measure would be to look at the shortest path length between different neurons. And if you take the inverse of that path length, that tells you how accessible these pair of neurons are, right? So if the path length is high, probably your signal will never make it to the other end, right? The noise will disrupt it. Now, if you take the average of this path length, you get a measure called communication efficiency. Now, obviously, you can ramp up the communication efficiency by simply, investing more wires, synapses. But then you have you have to trade off between the total expenditure of making this connectome and what you get out of it. So like a simple comparison would be to just look at the communication efficiency and the total process length that you have to use to make that connection. Now the C. elegans connectome, given that we know the connectome exactly, shows we can calculate both the parameter factors, and for comparison what we can do is we can just randomize these networks, preserving its degree, out degree and in degree, and we can ask how does these randomized networks behave, and you can show that what we observe is that the C. elegans network is suboptimal compared to randomized networks. So clearly it's not just designed to propagate signals from one end to the other. There is a lot of what could be the other reasons? I mean, we are under the assumption that neurons just want to access each other. We see that the nervous system is suboptimal. So what explains this suboptimality? What is the nervous system designed to do apart from just communication? It's a trivial question, it's not a trick question. I think a lot of you know the answers, but there's just, I mean, one answer, okay, I thought you would say specificity, but yeah, redundancy. So in the previous talk, someone pointed out specificity, right? You need to ensure that certain signals only go to certain regions. But another important factor is redundancy. You kill a particular neuron, somehow it's still able to perform its function, right? And usually, I mean, that's what generally separates a biological system from a, like let's say a computer, and this sort of redundancies and so. I'll just end with this question. There is no one answer, but I'll talk a bit more about function and development in the next talk. But maybe if someone wants to answer, how do you think are these things related? You don't have to look at all of it. Just let's just say structure and development. How do you think structure and development is related?
Through the status of development.
Right, like this.
Requirements, structure has to adapt.
Right, so you sort of assemble the network through development, right? Is there any other inverse relation, like structure somehow affects development? Can you think of any example?
An embryo has to be, there are structures that are formed when an embryo goes through gastrulation and that leads to proper development.
Okay, maybe in the context of connectomes. But yeah, that's a correct example. Yeah. I mean, one of the speakers did mention this, the fact that, so if you look at across different worms, there is some variability in the structure, right? But they're still able to do the same function. So is it something that depends on the history of its developmental trajectory? So in some sense, because their experiences were different as they were growing, they end up modulating the response slightly differently to different things. So it's like, the worm's personality and the variability in the connectome. I mean, it's an infinite regress. You can keep talking about this, but in the next lecture, I'll hopefully give some closure, as to how these things can be related and thought of. Any questions? Either it was too obvious or nothing made sense. Okay, fine. Thanks.